My name is Samuel Mauli Adadey. I'm presenting part of my PhD thesis focused on identifying genetic markers associated with non-syndromic hearing impairment in Ghana and also to develop an affordable and effective screening tool for a deafness gene in Ghana. As part of this presentation, I'll take you through a brief introduction, the rationale, objective methods, results and discussion, then the conclusion. Hearing impairment is the inability of an individual to perceive sound within the audible range of hearing. It is either caused by environmental factors or genetic factors. It is classified as conductive, sensorineural, or mixed based on the part of the ear that is affected. When the middle ear or the outer ear is affected, it's classified as conductive. It is classified as sensorineural when the inner ear is affected. When both the inner and the middle ear are affected, then it's classified as mixed hearing impairment. To begin the study, we conducted a literature search on hearing impairment publications from Ghana, and we identified that majority of the publications were on the environmental causes of hearing impairment, with only two papers on the genetics of hearing impairment, one from the eastern region and the other one which is a nationwide study. Both papers focused on connexin 26. Connexin 26, that's GJB2, is part of a family of proteins that form the gap junction protein. And this protein allows for cell communication and proper functioning of the ear. We found out that the previous studies on GJB2 identified a founder mutation within the Ghanaian population. Knowing that GJB2 accounts for a large proportion of hearing impairment cases in Ghana, and it's been about 18 years now since it was last studied in Ghana, we decided to revisit GJB2 as well as screen other gene mutations such as GJB4, GJB6, and GJC3 mutations in familial and non-familial cases in Ghana. We also developed a rapid and cost-effective tool for screening the founder mutation within the Ghanaian population. We visited 10 schools for the deaf across Ghana and identified children with hearing impairment. Majority of them had prelingual hearing impairment, that is, they developed the condition before the age of language development, with few of them having postlingual hearing impairment. With the prelingual hearing impairment, which is our focus, we classified them as familial cases where we obtain families with at least two affected individuals from the same family, and isolated cases where only one person is affected within the family but has a putative genetic background of the condition. We extracted DNA from them, used Sanger sequencing to screen for the connexin gene mutations, and did the sequence alignment and analysis. The results of our study identified a mutation within the GJB2 gene in a family, and this mutation results in the production of a truncated protein. And previous report shows that the truncated protein is non-functional and is likely responsible for the deafness in these patients. Further analysis of the GJB2 gene has shown that the previously reported founder mutation within the Ghanaian population remains the major genetic factor associated with hearing impairment in Ghana. It accounted for over a quarter of the familial cases that we screened, and it also accounted for about 8% of the isolated cases. Knowing that this mutation is highly prevalent within the Ghanaian population, we decided to investigate the carrier frequency of the mutation, and we found a carrier frequency of 1.4%. Our interest to develop a rapid diagnostic tool for screening the founder mutation within the Ghanaian population was motivated by the high carrier frequency of this founder mutation within the Ghanaian population. We designed the assay based on the restriction fragment length polymorphism test using the restriction enzyme NCI1. We amplified the coding region of the GJB2 gene that has the founder mutation, and we use a restriction enzyme to digest the PCR amplicon. We resolved it on 2% agarose gel, and we have distinct band patterns for the wild type, for the mutant, and for the heterozygote. These distinct band patterns help us to differentiate between the various genotypes present in our sample. We used the RFLP test to screen for the founder mutation within families that were recruited from Adamrabe. Adamrabe is a village in the eastern region known for its high prevalence of hearing impaired patients, and we found out that 7 out of 8 families that were screened had the founder mutation segregating with the hearing impairment phenotype. However, one of the family did not have the founder mutation segregating with the phenotype. All the tests that were conducted were validated with Sanger sequencing. In addition to the Adamrabe cases that were screened, we also screened isolated cases from the schools for the deaf that were included in this study. Using Sanger sequencing as the gold standard, we did not identify any false negative or false positive test. We also found our test to be 100% specific and 100% sensitive. However, the specificity is dependent on the mutations that are present within the recognition site of the restriction enzyme. The phenotype to genotype correlation has shown that all the patients with the founder mutation had profound hearing impairment. There was, however, no significant difference between the carriers and the heterozygotes normal for the founder mutation. In addition to the GJB2 screening, we also screened GJB4 and GJC3 and GJB6 for gene variants. We found a likely pathogenic gene variant within the GJB4 gene, and this likely pathogenic variant had the amino acid asparagine to be highly conserved at the position 119 in all the species that we studied. In addition, we wanted to find whether the variants would have any effect on the protein folding and the protein function. We found that it affected the protein structure such that the normal random coil that is formed at this position has changed into a helix, and the change in structure has also affected the binding property of this protein such that it binds an additional ligand which the wild type did not bind. In conclusion, GJB2 remains to be the major gene associated with hearing impairment in Ghana, and therefore it should be considered for the regular clinical investigations. And also, we advocate that the Ghana Health Service should consider using the RFLP test that we designed to screen for the founder mutation within the Ghanaian population, and also use this to complement the newborn hearing screening program in Ghana. We did not find any variants within the GJB6 and GJC3 region that significantly associates with hearing impairment within the Ghanaian population, and hence should not be included in a regular screening program in Ghana. I would like to use the opportunity to thank my supervisors, Professor Ambrose Wonkam, Professor Gordon Awandari, Professor Jeffrey Amedofo, Dr. Osborne Kwe, for their guidance during my PhD. And also, I wish to thank the people who funded this work, and all the staff and the head of the schools for the deaf, and all the parents and students who enrolled on this project. Thank you.